Hi everyone, now I'm going to talk about the new breakthrough that has come in gene editing, and I do not know why people are not talking about this more. So till now what gene editing has looked like is basically this method called CRISPR. What CRISPR does is that there is this protein called Cas9 which acts as a pair of molecular scissors. It is guided by a guide RNA which scientists create, which contains a cRNA which is basically the sequence that you want to cut off, and a tRNA which is basically a connector between the cRNA and the Cas9 protein. This is merged into a single guide RNA which guides the Cas9 protein to the part of the DNA that scientists want cut. This can be a faulty gene or it can be anything, and then this is cut. So this is cut. Now what happens after this is that either the genes, the parts like the two ends of the DNA, they either repair themselves or the scientists insert a new DNA sequence between that part. So what the issues that come with this are that either mutations can happen, like while repairing itself some letters can be rearranged. So the new breakthrough that has come is called epigenome gene editing. In this, what exactly happens is that on top of DNA, you place a protein that can turn off these chemical markers that activate certain parts of the DNA on and off, like you can activate or deactivate it. How this works is that DNA is basically a complete instruction manual. Like our skin cell and our muscle cells have the exact same DNA, but certain parts of the DNA, the genes are either on or off depending on the cell. So different parts are on in different cells. So what this epigenome editing does is basically it takes a dead Cas9 protein and it places another protein that can turn these chemical markers for certain genes on and off. So it's basically what this does is that you can turn off faulty genes as you want, and this is going to be way more accurate because gene mutations, etc., the chances of that happening are low. So this is right now being explored with FSHD by this company called EpiCRISPR Biotechnologies, and then Enchroma has proved that the cholesterol level in monkeys because of this reduced by 70%, and then also there is it being tried on chronic hepatitis B. Now this is still in its early clinical trials, but this is huge. The first generation of gene editing rewrote the code of life, but now the next generation of gene editing is going to leave the code untouched and simply tell our cells which part of the code they are allowed to read and which part they are not.